your busy schedule to join us this evening. We hope you'll learn today. We have lined up for you to be fruitful and engaging. God speaks in the silence of the heart. Listening is the beginning of prayer, says Mother Teresa. May I invite everyone to invoke the Almighty. Let us all bow down our heads for invocation.
There shall be showers of blessing, promise of love. There shall be seasons refreshing, sent from the Savior above. Showers of blessing we need. Mercy drops round us are falling, but for the showers we There shall be showers of blessing, precious reviving again, over the hills and the valleys, sound of abundance of rain, showers of blessing, showers of blessing we need. Mercy drops round us are falling, but for the showers we plead.
Susan Jeffers said, if the sign on your heart says welcome, the love will come pouring in from everywhere. To begin this program, we are pleased to have Dr. K. Rajan, Associate Professor of Zoology, St. Joseph's College, Tiruchirappalli, to deliver the welcome address.
Thank you, Dr. Anisha, for this wonderful opportunity. Yes, I am very happy to welcome all the participants. First and foremost, I would like to welcome our beloved Secretary, Reverend Father Dr. Peter S.J., who is always kind-hearted and very approachable and supportive to any type of academic venture, and also the college management for their constant encouragement and support. So on behalf of the Botany Department, all the participants, and on behalf of the organizing secretaries, we wholeheartedly welcome you, Father, to this webinar. Welcome, Father. I also welcome Dr. Sagaya Sadish, Dean, School of Biological Science, St. Joseph's College, who is always supportive with encouraging words. Dr. Sendal Kumar, the head of the Department of Botany, is our backbone for the success of the department growth and sustenance. And also all the faculty members and the students of Botany Department and other departments of St. Joseph's College, we are cordially welcome them to this webinar. Welcome, sir. Moreover, all the participants from various universities, colleges, and all the other institutions from India and other countries like Iraq, Hong Kong, Indonesia, Tunisia, Egypt, Algeria, Yemen, and Mexico. So, etc. On behalf of all the college management, Department of Botany, and on behalf of the organizing secretaries, we extend a warm welcome to this webinar. Welcome, sirs. I also welcome Dr. Kanickai Raj, Associate Professor of Physics, St. Joseph's College, for his untired and meticulous technical support for the smooth running of this webinar. Welcome, sir. Last but not least, we welcome today's man of this webinar, Dr. Vishnu Prabhu Dureraj Pandian, a scientist, University of Maryland, Baltimore School of Medicine, Baltimore, USA, going to deliver a talk on the topic genetics factors, the root cause of drug responsive variations. He will be introduced by Dr. Anushya later. At this juncture, even at this pandemic situation, he spared his valuable time for today's webinar. So on behalf of the college management, Department of Botany, all the participants, on behalf of the organizing secretaries, we are proud and happy to have you as a resource person to this webinar. Welcome, sirs. Once again, welcome everyone. Thank you for joining us today's webinar. Thank you.
Thank you, sir, for your kind words of welcome. May I now request Reverend Father Peter S.J., Secretary, St. Joseph's College, Tiruchirappalli, to say few words about this webinar and bless us.
Thank you, ma'am. It gives me great joy to greet you all warm and pleasant evening. I feel so happy and thankful to so many professors. Even during this COVID lockdown, they are working and organizing various seminars, national online seminars as well as webinars. This day is an auspicious day and a wonderful day for St. Joseph's College, Tiruchirappalli. The Department of Botany, one of the oldest departments, is organizing a very useful international webinar The Department of Botany, one of the oldest departments, is organizing a very useful international webinar on genetic factors, the root cause of drug response variations. Professor Dr. K. Rajan and his team, guided by Professor Dr. Sendil and Dr. Sahai Satish, have taken a lot of efforts to make this international webinar a relevant and useful one. Even though I am a social scientist belonging to the Faculty of Commerce, I could sense how important this international webinar could be. I wish all the participants pay greater attention in this interaction and expose. A word about international webinar. Genetic variations among different ethnic groups have been well established to associate with the risk of non-infectious diseases like cancer, CAD, CAD, autism, etc. Apart from the risk association, currently many SENPs have been functionally associated to be responsible for inter-individual variability during a course of drug therapy. Pharmacogenetics is the study of how people respond differently to drug therapy based upon their genetic makeup or genes. These pharmacogenetic single nucleotide polymorphisms are mainly of the drug transporters and CYP family genes, which are responsible for the varied drug effect among individuals and gives a range of responder category. Personalized medicine is the tailoring of medical treatment to the individual pharmacogenetic background of each patient. The approach relies on scientific breakthroughs in our understanding of how a person's unique molecular and genetic profile makes them susceptible to certain diseases. The webinar certainly will focus on the understanding the importance of genetic variants and how they impact the drug response variation among the individuals and the futuristic approach of therapeutic strategies implementing the pharmacogenetic knowledge. I am thrilled to see how as many as 1,000 participants have registered, and I am sure this webinar is going to be very informative as well as very useful. I wish the organizers, I wish the team all God's choicest blessings from above. Thank you.
Thank you, Father, for the wonderful remarks and your words of appreciation. Now, I'm happy to introduce the resource person, Dr. D. Vishnu Prabhu, postdoctoral researcher, Baltimore School of Medicine, University of Maryland, USA. Dr. D. Vishnu Prabhu obtained his BS Industrial Microbiology from Tyagaraja College, Madurai, and MSc in Biomedical Genetics from Madras University, Chennai. He earned his Doctor of Philosophy in Biomedical Genetics from the same university. His expertise in genetic analysis, linkage mapping, pharmacogenetics, and genomics. He's a genetics researcher with 4 years of postdoctoral research training in pharmacogenetics, cardiovascular genetics, and hypertension physiology. He has a rich research expertise as international pre-doctoral training in cancer biology, University of Naples Federico, Italy, has characterized the molecular signaling of CXCR4 activation in RET-PTC. He has worked as a project scientist, National Hub for Healthcare Instrumentation and Development at Anna University, Chennai. He was working as a postdoctoral fellow in Institute for Stem Cell Science and Regenerative Medicine, Bangalore, and in University of Toledo, Ohio, USA. He is currently working in University of Maryland, Baltimore in virology, especially on COVID-19. To his credit, he was a recipient of Madras University Merit Fellowship for PG. He has been awarded JRF by ICMR and SRF by CSIR and National Postdoctoral Fellowship from DST SERB India. Has published many research articles in reputed journals. Has characterized molecular signaling of CACNB2 gene, which is associated with hypertension. With his few words of introduction, I'd like to invite Dr. D. Vishnu Prabhu to proceed with his presentation. Now the session is yours, sir.
Thank you so much for your kind introduction, Dr. Anushya. And it's really a honor for me to present and share a little bit knowledge what I gathered throughout my course of education with all of you. And hope you're going to enjoy this presentation webinar, and it will be a useful topic for most of you guys. And hope everyone can listen to my voice and can see my slides. Okay, today's topic, genetic factors, the root cause of drug response variation. It's kind of little bit complicated topic because most of us feel genetics is a hard subject to understand, but it is one of the interesting topic to understand the genetics, and I will try to explain it in a very simpler way to everyone can understand. And the most importantly, this is called the futuristic medicine, and how the approach is going to change our therapeutic pattern and therapeutic response into the category of personalized medicine. And okay, so this is the outline of my today's talk. So I'm going to present you 5 concepts, 5 different chapters, as I have listed here. I'm going to explain about the genetics and the variations. So what are the basics about the genetics and variations, what are they and how they're playing a role in our physiological function and other factors. And the pharmacotherapeutics. So pharmacotherapeutics aspect, I'm going to talk the drug, when the drug is prescribed to a patient and what's happening inside the body and how they are going to help the patient to get rid of the disease. And pharmacogenetics, where we are going to combine these two factors, the genetic variations, what we already have in our genetic network, genetic system, I mean the genes in our body, and how the drug response is going to be impacted by these variations. That's the subject, pharmacogenetics. And I'm going to talk about the current trend of pharmacogenetics today. So pharmacogenetics, we are talking like it's coming in picture from for the last 3-4 decades. But what is the stage we are now? So I'm going to give you a few examples of how we are progressing in pharmacogenetics. And finally, I would like to show you what is a personalized medicine and what is a futuristic approach and how we are moving forward with this knowledge and which can be delivered into a clinical practice. Without any further delay, I'm going to start about the genetics and variation. Okay, it's very simple. Like we all know, human body is made up of organs, and these organs, they physiologically play a role to keep us healthy, and they all are made up of tissues. And these tissues, we all know it's made up of cells. So cells are the fundamental unit of life. And if you see the cells, there are a lot of intracellular organelles are there. I'm going to focus only on the nucleus because that's where our genetic information is is present and it's protected, and also it's passed to the next generation of cells. So when the cell divides, it copies the genetic information and it gives to the daughter cell. So to understand the genetics, we should know what is present in the nucleus. If you zoom into the nucleus, we know the genetic material is present inside the nucleus is DNA. As everyone knows, genetic material is deoxyribonucleic acid, which codes, which contains all the information about your genes, and it is present. It is highly compactly packed in the with the help of histone protein and forms a nucleosomes. And it's kind of winding a thread in a very compact way, and then this nucleosome assembly like again wound in another structural configuration to get a solenoid structure, and this solenoid structure forms a chromatin, and the chromatin forms a chromatid, which is an individual arm of each chromosomes. And finally, the compact form of chromosome, you can see in this picture in the metaphase, they have different distinct and unique structures. As everyone know, humans have 23 pairs of chromosomes. So 22 non-sex chromosomes, that's otherwise called autosomes, and one pair of sex chromosome, either XX in female, XY in male. So what is there in the genes? What is there in the chromosome? The most important fact is it carries all the information your cell needs. For example, I will give you a very brief example of what's happening. For example, insulin is a protein. Everyone knows it's produced in the beta cells of pancreas. The insulin coding gene is present in the genes in the chromosome in throughout the body. If you take the neuronal cells, there will be insulin coding gene in the nucleus of the neurons, but the insulin will be produced or expressed only in the beta cells, not in the neurons. In the same way, if you see the cardiac myosin proteins, which is present only in cardiomyocytes, they are the muscle cells of the heart. They only express in the cardiomyocytes. They are not going to be expressed in your skeletal muscles, or it's not going to be expressed in your liver cells. The concept I'm coming to say is the gene expression is highly regulated and it's tightly controlled by various factors, and that is called spatio-temporal gene expression. Spatio means time, at the location where at which specific type of cells the gene should be and produce the protein product, and the temporal, the time, what time it needs to be produced. So everything is highly controlled. It is kind of more fascinating to know about the gene expression regulation. And now we know like what is present in the nucleus, and we wanted to know more details about the genome. In this slide, I made a genetic analogy. It's a comparison. If you take the whole genome in a cell, that means the total amount of the sequence of DNA in the 23 pairs of chromosome in a cell compared to a book, each chapter we can consider it as a chromosomes. So totally the book of human genome contains 23 chapters. But the interesting fact is you will have each chapter a double copies because you are inheriting two chromosomes of the same. For example, chromosome number one, you will inherit one from your father, one from your mother. That's paternal inheritance. So the chromosomes can be compared to the chapter of this book, and the gene, if you want to compare the gene in the textbook, you can compare as a sentence. So a chapter will have multiple sentences. The same way the chromosome will have multiple genes. And a code, as everyone know, the genetic code dictionary is a triplet word. It's the starting codon is ATG, which codes for methionine. So the codon is a word, and the alphabets in the language is only four. That's adenine, thymine, guanine, and cytosine. And finally, the DNA is our language. So this analogy will give you an idea how we can able to understand the genetics and genes present in the chromosomes and the total genome. And in order to know the composition of the human genome is sequenced in 2003 by the Human Genome Project, and they curated approximately 3 billion base pairs. That's total amount of DNA we have is 3 billion base pairs. So the chromosomes are designated the numbers based on their descending order on the size from chromosome 1 to 22, but there is a misnomer. Chromosome 21 is the smallest chromosome, but 22 has been identified later. So the average size of chromosome is 150 megabases, and the genes are present in the chromosome. Average size of a gene 50,000 base pairs. I'm giving an average. There are different size of genes, like the smallest gene and the longest gene. So they are different, and each responsible for the protein product which it needs to do the function. And in the gene, we do have two different segments. One is exons, another one is introns. The exons, it codes for the amino acids, and introns are intervening sequences which is playing a major role in regulating the gene expression, and also they are non-coding sequences. And usually the exons are the sequence which codes for the amino acid coming out of this DNA, after the transcription, and produce a protein product. An average size of an exon is 3 kilobases. As we know, the genetic code is 3 base pairs. And most importantly, the effect of this genome is modulated by a single base pair variations. We call them as mutation or polymorphism. So we have 3 billion base pairs, and a single change in this 3 billion base pair at any point of location may lead to a genetic association or a disease. I would like to emphasize what is the difference between mutation and a polymorphism. So, as everyone know, mutation is a sudden genetic change at a single nucleotide level, as a base pair level. For example, adenine changed to thymine, or cytosine changed to guanine. So, this mutation, when happens mostly in the exonic regions where it codes for an amino acid and leads to a truncated protein or a functioning protein, directly resulting in a disease. So all the patients who carry this specific genetic variation, they may end up in developing the disease at any point of time in their life. But polymorphism is the same genetic change, but it doesn't cause the disease right away. But the polymorphisms, the same genetic change, may have an impact later. The patients or the people, they'll be normal in healthy, but in a later time, they may develop a disease. So it can be a disease risk associated variation. Additionally, this variation may influence the drug response variation and other factors. So we call them a single nucleotide polymorphism. So that's what I'm going to talk in my future slides. And more interestingly, like how many protein coding genes we have in the human genome. Initially, it was or more protein coding genes, but later, like scientists ruled out pseudo genes and non-coding genes. Something approximately we have 23,000 protein coding genes, which helps to keep the cellular function normally, and also it produces essential function of the cell. Okay, and one more concept we need to understand in genetics, that's an allele. So allele is one or two or more alternative forms of a gene. So as we know, like most of us, we studied the genetics, like Mendelian genetics, like dominant allele or recessive allele. So it just represents a wild type allele, which is a normal gene, and a mutant or polymorphic allele, as I mentioned here, they will be a defective allele. So, and interestingly, you will get two copies of gene from your family, maternal and paternal. So, ultimately, it may be a homozygous or heterozygous for in the case of allele. So, you may have both wild type alleles of gene. Otherwise, you may have one mutant or polymorphic allele and another one will be a wild type. In that case, you'll be considered as a heterozygous. So, and these concepts are very essential to understand the further slides in which is I'm going to explain to you. So I'm going to give you a small trivia, gene trivia. What is an interesting fact about the genetics? Which is the longest gene in human? It's DMD. It's Duchenne muscular dystrophy gene, which has been known to cause the disease of Duchenne muscular dystrophy, and is a protein called dystrophin. It is 2.3 megabases in size. It's approximately 0.08% of the human genome. Imagine how big it is. And another interesting factor is it takes 6 hours to transcribe the DNA to mRNA, which is the intermediate nucleotide element, which helps to make this as a protein product. So it takes years to complete one copy of this protein. So this is kind of more fascinating to us to know, like genetics is highly complex. In the meantime, is highly global. Okay. Now we know what is genetics and what are the genes playing a role and how they are the single nucleated variations happening in our human genes. So in this slide, you can see the list of genetic variations happening at the single nucleated level. In the single nucleated level. So the first one is substitution. Substitution, you can see the wild type in the top. They have a T, that's thymine, in the wild type, replaced by an insertion is like they insert cytosine in the front of thymine in a mutant. And deletion is removing the thymine here. You can see that is a mutant. And indel, indels are insertion deletions. Insertion like you in the wild type TG, but the G is removed. That's deleted here, and there is a C inserted. So these are single nucleotide variations you can observe throughout the genome. So the copy number variations, which are a long stretch of DNA sequence variation, locations and numerical changes like a new, but maybe a different session. But mostly, I'm going to focus on the single nucleotide variations, which has been considered as a most important category in the pharmacogenetics and responsible for drug response variation. So if you see the human to human genetic variation, so if you take as an Asian population, Indian population, us, if you compare our whole genome with a European or any other population like African population, you cannot expect more than 99.5% similarity. You can expect like only 0.5% difference. So even though like we are far distinct from ethnicity, we are very close to their genetics, but impact in causing that should be 0.1% or even less. But 0.1% so high. So as of 2019, in 2019, the nucleotide polymorphism database, and the build is 153, listed 675 million variants found in various human genomes, like not only restricted to Europeans, also Asians and other populations. Out of the 670 million, you cannot say all of them have an impact in your physiological function or metabolic function. There are uh, thousands and thousands of variants play a major role in modulating your drug response variation and other genetic biomarkers, I mean, predisposing you to get a disease. So now we know what is a genetics and what is a variation, how they are, so especially the single nucleotide variations. Now I need to give a brief uh, understanding of pharmacotherapeutics. I'm not a pharmacist, I can explain it better, but I can able to explain it in a simple way. What's happening after you prescribe a drug? So the ultimate goal of medication or pharmacotherapeutics is improving the patient's health. When the drug is prescribed, it is administered either orally or intravenous, and mode of administration differs between the drug. And the administration after that, it has the drug will be absorbed in your gut, especially in the small intestine, and they go with the metabolism and activation. And this makes the bioavailability of the drug. So the bioavailability makes the active drug to reach the peak plasma concentration, and then they reach the target tissue where it needs to be functionally working, and it binds to the specific type of receptor or enzyme or whatever, and it shows the physiological effect. Then it improves the patient's health. And in the meantime, when you have the more amount of active drug in the body, most of the drugs which is in the circulation are toxic to the patient. So the body, what it does, it clears the drug by metabolizing them or breaking them down and excrete them via like urine, or it can be via like even fecal matter. So the pharmacokinetics and dynamics plays a major role in making the drug available to the patient and improving the patient health. Pharmacokinetics is nothing but what the body does to a drug. So in simple words, you are taking a drug and how your body is going to break it and make it bioavailable and excrete it out of your body. This is pharmacokinetics. And pharmacodynamics is what the drug does to does to our body. So after it reaches a peak plasma concentration, how it binds to the target tissue, how it's interact with the protein of its target, how it's going to give the effect on the patient. So this part is pharmacodynamics. So this is very simple, but the most important thing is all this the cycle involved in plenty of genes and proteins. That is what the concept. So all these proteins in various steps, the absorption, metabolism, and the molecular level targets of this specific drug, all of these genes produce a protein. They all have some genetic variation, which may impact at any point of the steps. That will lead to a impaired patient health. So that is what we are going to talk. So I'm going to give you a brief you two important categories of the uh, genes and families, which plays a major role in pharmacokinetics. And first one is drug absorption and transporter genes. So we know like that once you take the drug orally, it just get absorbed in your small intestine, and most of the drugs are absorbed in the small intestine. The drug transporters are highly expressed in the small intestine, and two major category superfamily are there. One is the ABC superfamily and the solute carrier superfamily. So ABC is nothing but ATP binding cassette. So it needs an energy ATP molecule to take up the drug inside the cell, as well as it pumps out the cell. So they play both roles, like taking inside the cell and also pumping out the cell. So most of you know, like the multi-drug resistance genes or the proteins are belonging to the ABC superfamily. And solute carrier superfamily are another major superfamily of proteins involved in taking up the drug inside the body, inside the cell, and also pumping out. So the variations in these genes can modulate the function of drug uptake. For example, if you are given 100 milligram of drug X, whatever, and this drug, when you have genetic variation in the specific protein which involves in taking up the drug inside the body, then there will be a defect in taking up the drug inside your body, then it will not give you a positive result in the therapeutic outcome. And if you see the number of genes, the ABC, it has 48 different genes, and it codes for more than 200 different transporter proteins. And it is expressed in small intestinal epithelial cells, also it in the liver, in various other tissues. And simultaneously, SLC, the solute carrier protein superfamily, have 65 gene families produce more than 400 different transporter proteins. So a specific drug will be transported majorly by any one of the protein, also additionally some of the other family proteins. So the concept, the point I'm coming to convince you here, the variations are large. You can see 400 different proteins, they may have different variations, they may have different impacts. So we don't know what will impact your drug uptake. But we have some solid evidence that I'm going to show you in my coming slides, how they are going to impact. So here in this slide, I'm giving you a very brief example. Here you can see the effect of the genetic variation in the uptake of a drug and effects of a drug. You can see this is inside the cell. It may be an intestinal epithelial cell, hepatocyte. You can see the arrow marker, the thickness, the intensity of the arrow is higher here, it's lower here. So that represents the variation in the SLC. I mean, these are the genetic variations in the specific SLC family protein. If you carry this, the uptake of the drug will be low. This one is in influx. That means taking out from the bloodstream to the cells. In the same way, the variation in this ABC family in the efflux, pumping out the drug from your cell will be low. That means accumulation of the drug inside the cell will be for a long time. Here, uptake will be low. You not get for your physiological effect. The alternate, when you have specific genetic variation in this SLC family of protein, you will pump out. So here, the category is in this case, like you'll have more amount of active drug, and the ABCG2 here, it pumps out this variation, pumps out the drug effectively from the cell to the bloodstream. So now you understand, this is just a very brief example to make you understand how this genetic variations in this transporter proteins influence the drug and efflux. Now, the second most category in the drug response variation is cytochrome P450 families. They are the pioneer, they are the primary enzymes in metabolizing all the which you are taking inside your body. And the Human Genome Project, like we identified 57 human genes coding for various cytochrome P450. This kind of enzymes can able to metabolize like 75% of all the drugs and chemicals or xenobiotics which is coming inside your gut, inside your bloodstream. Therapeutic drugs, like 70 to 80% therapeutic drugs which is in clinical use are metabolized by cytochrome P450 family 1, 2, 3. So this only 3 families of protein are responsible for biotransformation of most of the foreign substances. And one more important concept and point we need to understand here. You may take a drug, a active drug. So what is a prodrug and active drug? Prodrug is a drug which you need to be broken down and undergo a biotransformation to become an active drug. In other words, if you are taking an active drug which doesn't need a bioactivation, and it and later that drug will be broken down and eliminated from the body by the CYP. So the CYP plays 2 major roles: one for biotransforming an active drug, and also an active drug once it's done, you cannot have it a long time in your circulation in your blood. That may cause some side effects. So it clears out from your body and you rid of it. So that's what I mentioned here. And I'm going to give you interesting information about this enzyme, cytochrome P450. So P450 has a name P450, what it stands for. Actually, when the enzyme was identified, it was named as a color compound because chrome means color. Everyone knows chrome. So containing enzyme. When they have spectrophotometry, they had an absorption maxima. They have named it as cytochrome P450. But later they identified this role in metabolizing the drugs. A pie chart example. So you can see the different families of enzymes. And you can see these are, for example, their efficacy. How effectively this breakdown or metabolized drug is influenced by your age, or influenced also by other factors like environmental factors. Some of them they have a genetic background. The polymorphism listed here they plays a major role along with other factors in order to show a variability in the drug response among the patient. Okay. Now we know that the metabolism is highly influenced by the cytochrome P450 genes, and proteins also plays a major role in modulating response drug metabolism effects. So we can able to categorize patients based on makeup. For example, here you have normal. So normal allele in the sense they are normal gene, functionally normal genes. So those patients they have been given a specific drug, they will be an extensive metabolic. They will be the best to respond to the treatment. But there are some patients will have a duplicated gene, duplicated in the sense they may have an increased activity. They will be ultra rapid metabolizers. When you give a drug, they develop the drug so quickly, and there is a problem with that because you don't have the drug in the system for a long time because they live very short in your biology, in your blood plasma. So you may end up in switching to an alternative therapy, or maybe you need to go for a multiple dose for the patients, those who carry the duplicated genes or the gene which has a genetic variation which has an increased enzyme activity. And now we come to the interesting point, like partially defective allele, what you see here with a red triangle. So some of the patients may have a partially defective, that means only 50% of enzyme activity you will be having. Those will have only 50% of therapeutic results. They will be called intermediate metabolizers. So these patients may need a higher dose of drug, or they need a constant monitoring for their therapeutic outcome, or they may need an alternative therapeutic strategy. And the final stage comes when you have a null allele. Null is nothing but complete loss of function of that specific enzyme due to the genetic variation. They are going to be considered as a poor metabolizers. They are not going to respond to the therapy since the role of this enzyme is completely lost. So the enzyme cannot metabolize the drug, and they will not be having any therapeutic benefit out of this drug. And these patients definitely need an alternative therapy, alternative medication in order to achieve the therapeutic outcome. Now you understand how we classify the patients based on their genetic makeup and their metabolizing potential. In simple words, now I am going to give you a Punnett square. So Punnett square is nothing but making a combination of events, how it can give the impact, the clinical impact. So I'm going to take the case of cytochrome P450 enzyme. You may have a loss of function variant or a gain of function variant. The drug you're going to introduce into your body may be an active drug or prodrug. So we will go a case by case. When you're taking an active drug, if the patient carrying a loss of function, that means the enzyme lost its function. The active drug needs to be cleared out from the body because mostly they may be toxic to the body, but the loss of function enzyme cannot clear the active drug in your system. Also, the plasma concentration will be higher. The chances of getting side effects in this patient will be so high. So technically, they need to go for an alternative medication or alternative drug, which doesn't involve in the specific CYP enzyme that's another CYP enzyme can metabolize it effectively. And coming to the gain of function category, the active drug, when you have an active enzyme, I mean active than normal, the clearance rate will be higher and the plasma concentration will be dropped so soon. That means if you expect a drug to be in your body for 24 hours, in this patient, it will be cleared out in 6 hours or even soon. In that case, you may go for multiple dosage in a single day, or you may go for an alternative therapy which can able to sustain for the 24 hours of therapeutic recommendation. Now coming to the pro-drug category. I mentioned already pro-drug which needs active metabolism to convert them into an active drug and which can lead to increased bioavailability. Now we have the category of loss of function. I mentioned pro-drug needs to be biotransformed to an active drug. But when you have loss of function allele, the bioavailability of this prodrug, the active form of drug will be low because that is not broken down into an active form. Obviously, these patients with a loss of function enzyme and the prodrug will not be benefited out of the treatment. And the same another fact, like you can see the gain of function allele, they will be effectively converting the prodrug into active drug, and they will be working better. But also we need to keep them in monitor in order to find out is there any side effects with these patients because of high bioavailability of the active drug in the plasma. So this Punnett square will give you like an idea how we can able to classify patients and identify. So this loss of function, gain of function alleles I'm talking about, they will be unique for specific enzymes. So if you find an alternative drug which is not influenced by this enzyme, then they can be recommended for the alternative therapy. Okay, so now we understand what the pharmacokinetics and dynamic genes plays important role. Now I'm going to extend my talk with the pharmacogenetics. So we understand genetics and variation, and the second chapter we understand what is the major role of the drug transporters and the drug metabolizing enzyme CYPs, and how the genetic variations are going to impact your therapeutic outcome. So now I'm going to start talking about pharmacogenetics. Let's start with the history. When this thing started, everyone knows Pythagoras. Everyone, because all of us studied Pythagoras theorem, one of the very interesting theorem like we studied in our school. In 510 BC, Pythagoras and his team, they have encountered some of the people when they have eaten fava beans. Fava beans are a kind of beans, like it's similar to, it's called special beans, and this is a mostly Eastern, I mean, the Mediterranean diet and also the European diet. So some of the people, those who take the fava beans, they've been triggered to acute hemolytic anemia. That's called favism. So what is favism? That is the patient, the people, they don't know what's going to happen in their body, but they develop acute hemolytic anemia after they take the fava beans. What happens? They break down your red blood cells and they turn them into yellow, the skin yellow, and their urine turns dark because of the heme content is released in your urine. But they don't know what is the mechanism, what is the genetic background behind this. Later, they found out it's because of the G6PD, that's glucose 6-phosphate dehydrogenase deficiency, which is an X-linked recessive disorder. But they could able to find out the people, some people will respond to drugs, like we call them like drug allergies or drug responses, because in ancient days, food was our drug. We have used the food as our drug to have a healthy life. So the recording the individual variety in responding to the food has been documented back in 510 BC. Understanding the molecular later. And the history continues in 1956. The patients, those who have been administered with a muscle relaxant called succinylcholine, some of them have shown prolonged paralysis after administration. Only a few of them. So that means they have some different side effects, but they could not understand what is the reason behind. And the British Medical Journal in 1960, they published the antibiotic used to treat the tuberculosis, isoniazid. Some of the patients, they have defective metabolism, so they don't respond well to the treatment. And in 1955, the very famous anti-malarial drug, now everyone knows, the hydroxychloroquine, the same family, primaquine, is administered to African American patients. And during the World War II period, they have encountered hemolytic anemia in the patients. And later they found they are also G6PD deficient. So now we understand, like everyone now interested in taking hydroxychloroquine. But if you have a G6PD deficiency, the FDA gives a warning. The Food and Drug Administration Safety Board in USA, they have given a warning. If you have a G6PD, that's glucose 6-phosphate dehydrogenase deficiency, you may get a hemolytic anemia. That's one of the labeled warning. And as I mentioned previously, the genetic polymorphisms in any category of genes which plays a major role in pharmacokinetics or pharmacodynamics, potentially influence your drug response variation and adverse drug reactions. That means side effects. So it's very clear not all the patients respond to the drug therapy in a uniform way and a beneficial fashion. The pharmacogenetics is a study how people respond differently to the drug therapy based on their genetic makeup. So that's what the concept we are understanding now. Now we know the importance of the genetic variation and the drug response variation, how they are very important to study and in developing a future medicine. And in this table, I'm giving you a very brief example. There are various drugs which is used to treat various systems like skin system, gastrointestinal, central nervous system, hematological, cardiovascular, even cancer biology. All of them, they have adverse drug reaction. So they have a strong genetic association. So this is a very basic glimpse. The picture continues to grow, and the scientists identifying novel variations strongly associated with drug response variation and or adverse drug reactions in various populations. And another important fact we need to understand, all these genetic variations are very specific to population. For example, you can see this, the star 2, star 3, they are kind of, their nomenclature, they name them to identify these variations. And these variations will not be present in all the population. For example, the star 2, here, what I see is shown here. They will be mostly present in Asian population. If you go to the European population, the frequency of this variation will be low. So the gene frequency and genotype frequency are going to play a major role in order to maintain the drug response variation. For example, you may be effectively treated by a drug since you belong to an Asian ethnic patient, but the same drug will have a side effect in a European patient who has a different genetic background. So that is more important to understand in this, in order to understand the pharmacogenetics. And now, in a better way, like as I mentioned, 90% of the population. So, for example, if you take my genetic network and compare with you, or even I compare with my wife or my parents, there may be a difference. At least we may have one genetic variation that can affect how our body is going to break down a medicine. But you are going to be fortunate if you are not going to take the drug if you have a bad gene. But some of them, like, they might have to take a medicine, but ultimately if they carry a defective allele, a defective gene or genetic variation which may lead to an adverse reaction, they may end up in developing an adverse reaction. So in this picture, I'll just explain it in a simple way. For example, this medicine what given is 100 milligram of dose. Based on your genetic makeup and other environmental influences, some patients, like, they respond well to the 100 milligram of drug without any side effects. They are based, the genetic makeup makes them to show better treatment outcome. Some of them carry an activated loss, gain of function variant, which they need only 50 milligram of drug. So they don't need to pump 100 milligram of drug, which a patient need only 50 milligram of drug. And alternatively, you can see there is one more patient, the 100 milligram of drug doesn't work because his genetic makeup makes the allele weaker, the enzyme weaker, or a kind of intermediate metabolizer. They may need a double dose, 200 milligram of drug to the response. And the most important category that we are going to be more concerned about, patients have a genetic variation that doesn't allow them to metabolize the drug or transport the drug into them, or also the target tissue of this drug might be having a genetic variation which makes them non-responders. In that case, you may need to go for an alternative medication. So this is very simple, and it's going to be like looks more complicated, but the concept is very simple. The basics of genetics, if you understand, and how to implement in the clinical practice, that's how the beauty comes of personalized medicine. And now we are going to watch a video, and that's going to give you an idea.
Pharmacogenetics refers to the study of genetic influences on an individual's response to drugs. In pharmacogenetics, the analysis of a specific gene or group of genes may be used to predict responses to a specific drug or class of drugs. Pharmacogenomics refers collectively to all the genes that influence drug responses and how genome-wide analysis may be used to identify such genes in the search for novel drug targets and/or key determinants of drug reactions. The effects of a specific dose of a specific drug will differ between individual recipients. A drug that is effective in one person may have no discernible therapeutic effect in another, whilst a third might show a partial response. In some, there may be undesirable side effects. There are multiple contributory factors to such variation in drug response, such as gender, age, body mass, diet, the presence of other drugs, or of particular disease states, and exposure to certain chemicals or toxins, such as cigarette smoke. In addition to these, genetic factors also influence drug response. Genetic factors may account for between 20 to 95% of the observed variation in drug response between individuals. Drugs undergo 5 stages following administration. 1. Absorption into the body, for example, from the gut. 2. Distribution to their site of action in the body. 3. Target interaction, for example, binding to cellular receptors or ion channels. 4. Metabolic processing. And 5. Excretion from the body. Theoretically, genes that influence any stage of this pathway could affect the overall drug response. There are 2 main groups of genes that are important when studying variation in drug responses. The first are those influencing the pharmacokinetic properties of drugs, such as drug metabolizing enzymes and drug transporters, which affect how the drug is handled by the body. The second is those influencing pharmacodynamic properties of drugs, including drug targets such as enzymes, receptors, and ion channels, and their associated pathways which determine the drug's effect on the body. One example of a gene that affects pharmacokinetic drug properties is the ABCB1 gene, which encodes the NDR1 drug transporter. Variants of ABCB1 are associated with resistance to the effects of drugs, such as the antiepileptic agent phenytoin. One example of a gene that affects pharmacodynamic drug properties is the CYP2C19 gene, which encodes the metabolic enzyme cytochrome P450. Variants of CYP2C19 are associated with a decreased level of responsiveness to omeprazole, used to treat peptic ulcers and other gastric complaints. Most of the genetic variants that affect drug action are called single nucleotide polymorphisms, SNPs, or sets of closely linked SNPs called haplotypes. SNPs are commonly occurring single nucleotide substitutions of one base for another, present in at least 1% of the general population. However, haplotypes are generally better predictors of drug responsiveness than isolated SNPs. For example, if responsiveness to a particular drug Y is affected by a set of SNPs in gene X, and there are 5 commonly occurring haplotypes, combinations of these SNPs in a population, then 2 might be associated with good responses, 1 with moderately good responses, and 2 with poor responses. Thus, using real-life illustrations, there are 2 common variants, C or T, at a specific position in the multi-drug transporter gene ABCB1. Most individuals will have the genotype CC, CT, or TT with respect to this SNP, and the CC genotype has been associated with resistance to antiepileptic drugs. Efficacy and toxicity are 2 key aspects of drug response of clinical relevance. Efficacy refers to the maximum beneficial or therapeutic response that a drug can produce, and is a measure of clinical effectiveness. It can be expressed in terms of the percentage of recipients who show a therapeutic response at a given standard dose. Toxicity refers to the extent to which a drug induces unwanted or harmful health effects, and may be expressed as the percentage of patients who show adverse side effects at a given dose. The optimal dose range for a drug is that at which efficacy is greatest and toxicity is lowest. In practice, achieving an acceptable level of toxicity usually means that efficacy is compromised, and in some cases, even the safest drugs can produce severe side effects. Extreme drug responders within a patient population are of concern. These include non-responders, in whom the drug is ineffective, and adverse responders, in whom the drug causes major harmful side effects. If it was possible to identify these individuals in advance, treatments could be improved. The non-responders could receive an increased dose or alternative medication, and the adverse responders could receive either a greatly decreased dose or an alternative form of medication.
Okay. Now, that video gives a very clean understanding of pharmacogenetics and how it works. And now I would like to move on to the current trend. What is happening right now? Now we know like genetics and how they play important role, the genetic variants in drug response variation. And what is the current knowledge and what is the current trend we have? And I would like to share you about one of the important organization called CPIC, that's Clinical Pharmacogenetics Implementation Consortium. So this organization is a non-profit private organization, autonomous body. They started in 2009, and they started to investigate the genetic variations of this cytochrome P450 family genes and other transporters, and how they can potentially modulate the drug response. And their goal is to address the clinical implementation of this specific genetic variations, which leads to a pharmacogenetic test, and to develop some guidelines how they can interpret and how this can be implemented for clinical practice. For example, as already we've seen, the metabolizer category, if you are a poor metabolizer, then they should give a guidelines. The patient should be preferably go with an alternative therapy. If the patients are going to develop an adverse reaction based on the genetic makeup, they will develop a guideline to give a brief idea or some advice to the clinicians how they have to handle the therapeutic response. And they also publish the guidelines in the PubMed database. Also, they are highly endorsed by various pharmacological societies in America. Also, the genetic system like clinical genetics. So something, there is a database ClinGen where they curate and deposit only the genetic variants which has a clinical impact. And PharmGKB is a pharmacogenetic genome knowledge base website. So up to date, like you have at least 250 prescription medications, what we are taking day to day. They contain a pharmacogenetic information which has their FDA approved labels. So now it starts from few decades before. Now we are in a stage to understand how your genetic makeup can modulate your drug response, at least for 250 prescription. And the figures and the numbers are going up because we are evaluating and estimating the impact of this genetic variation in various research labs, and you mean just finding out how they are going to be impacting our therapeutic response. And in this, I'm going to give you two examples of two interesting drugs. Since I'm coming from a cardiovascular background, I'm going to take two important drugs which is commonly used to treat cardiovascular patients, those who have a sudden cardiac arrest or obesity. And the first drug is clopidogrel. Clopidogrel is a drug which is an antiplatelet drug, which is an antithrombotic drug, which is given for patients though to prevent atherothrombotic events. I mean, simply to say, to prevent the blood clot inside your blood. Why? Because when you have a sudden cardiac arrest, patients, those who have a restricted blood flow in their coronary artery, the artery which supplies blood to your heart, there will be a procedure called percutaneous coronary intervention. They implant a stent in your coronary artery to 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 just get the blood supply back into the coronary artery. And the stent is the metal particle, which is a foreign particle. So obviously, like, your body will sense it as a foreign particle, and your platelets will be triggered to form a thrombus. So in order to stop the instant thrombosis, the patients will need to take 75 milligram of clopidogrel every day. It's more than 40 million patients worldwide taking this drug for their lifelong. And most importantly, this is a prodrug. As I mentioned, this prodrug needs to be bioactivated by the cytochrome P450 and primarily done by this enzyme, cytochrome P450-2C19. And scientists found out some genetic variation which can impact the metabolism of this prodrug. So they have named them as star 1, star 2, star 3, and star 17. There are more than 20 different variants, but I'm going to talk only about this 4. They are more common or frequent genetic variations observed in the patient population. So star 1 is a wild type. Wild type, as I said, they are normal allele. They don't have any genetic variation. So these patients carrying this wild type, they can be effectively treated with a 75 milligram of drug. But the loss of function variants, the star 2, star 3. So I already mentioned these genetic variations. You can see what is star 2 here. So this RS number, what you see here, that is how they nomenclature. The nomenclature of SNP is like this. It starts with RS, and you can find out about more information about this variation in the SNP database in NCBI. And this star 2 and star 3, they cause an decreased protein product. And if you see the star 17, that's another genetic variation which has an increased protein product. That's a gain of function variant. So now we have two concepts. Like if you have a loss of function, either star 2 or star 3, the prodrug will not be metabolized, and you may end up in developing a stent thrombosis because the drug is not working for you. In star 17, the drug will be effectively metabolized into an active drug, and most of the patients, they are okay, but in future they may need more amount of drug since the drug is cleared from your body so soon because you needed that for 24 hours. So I'm going to show you a video like you can see and have. Okay. So now we understand like how to classify the patients based on their metabolic category and how to approach their therapeutic augmentation. For example, if you are a poor metabolizer, switch to an alternate therapy. The picture you are seeing in this slide right now, it's a labeled warning given by FDA in all the prescription boxes, the drug you receive at the pharmacist or the pharmacist at the pharmacy, like the clopidogrel, they give an alert to the doctor as well as the patient. The metabolism is highly influenced by the genetic variation of the CYP2C19. And the interesting fact is here, patients will not get a bleeding complication because this is a prodrug. Ultimately, the poor metabolizers may not show the better treatment outcome, but they are not going to get the side effect like bleeding. But we do have another drug called warfarin, which is another antiplatelet drug, which is a active drug. And this is also a prodrug, but it's actively causing a bleeding disorder. It's otherwise called acinocomarin. And the pharmacogenetics of that drug has very serious implication. The patient should be tested for some of the genetics like CYP2C9, that's another cytochrome P450 family, before administering the drug to make sure what is the potential of the metabolism of in the patient. So now it's very clear the patients need to know how the therapeutic strategy works based on their genetic network. And it's not mandatory to test them before administering the drug, but they will monitor their response. If they don't develop a good therapeutic response, obviously they will go and do the testing, genotype testing, genetic testing, and switch them to an alternative therapy. There are other alternative antiplatelet drugs that are available, which is not influenced by this gene, So they may be working better for those patients. Okay. And I'm going to show you the next video. The second interesting drug is statin.
Statins are a group of medications that reduce cholesterol levels. They were discovered by Akira Endo in 1971, a Japanese biochemist. There are several statins in the market currently, which are very easy to remember because they all end with the suffix statin. Cholesterol is produced by the liver and transported by lipoproteins, which consist of two types: low-density lipoprotein, which is known as bad cholesterol. Its main function is to transport cholesterol throughout the body. The other type is high-density lipoprotein, also known as a good cholesterol. This type of lipoprotein removes the excess cholesterol and carries it back to the liver for degradation. So how does it work? Statins work by competitively inhibiting HMG coenzyme A reductase. This enzyme catalyzes the reduction of HMG coenzyme to mevalonate, which is the rate-limiting step in hepatic cholesterol biosynthesis. Inhibition of this enzyme decreases the cholesterol synthesis, leading to an upregulation in LDL receptors in the liver. This increases LDL uptake by hepatocytes, decreasing the amount of LDL cholesterol in the blood. Who should statins be given to? According to the American College of Cardiology, statins are given as a prevention of cardiovascular diseases. In patients with LDL cholesterol more than 190 mg per deciliter. On average, statins can lower blood LDL by 70 mg per deciliter, which translates into an estimated 60% decrease in the number of cardiac events like heart attacks or sudden cardiac deaths. Contraindications of statins. Being drugs that are metabolized and excreted by the liver, statins are contraindicated in cases of chronic liver disease or elevated serum transaminases. Statins are also not given in pregnancy or lactation as they are in category X. Lifestyle modifications are strongly advised in patients on statins. These modifications include less caloric intake, low animal fat diet, and physical exercises. Monitoring creatine kinase is important in patients taking statins, as myopathy is one of the most common and dangerous adverse effects of this class of medications. Thank you.
Okay. So the second important drug, as I mentioned after the clopidogrel, is statin. So this statin, as you've seen in the video, it is one of the primary drug recommended by American College of Cardiology to treat the cardiovascular disease by keeping the LDL levels in a check. But if you remember the last piece of note in that video, the monitoring, the creatinine kinase levels shoot up in your plasma in some of the patients. So you had to keep monitoring the patients. That may lead to a myopathy. Myopathy is a muscle pain, and it's a dynamic process. Like starts from a muscle pain and ends up in rhabdomyolysis. Rhabdomyolysis is complete loss of muscles. That is due to genetic variations in SLCO1B1, as I have mentioned. This is a transporter protein which helps to uptake the statin into the liver. So the table, what you're seeing here, this is a haplotype, the genetic variation star 1A, star 1B, 5, and 15. These are the genetic variation of this SLCO1B1, the SLC family transporters involved in uptaking the statin into the hepatic system. So the concept is, when you have statin in the circulation for a long time, they potentially can lead to a muscle damage. So the import of the clearance of the drug from the blood into the hepatic system saves you by avoiding the overexposure of the statin in your body. So the wild type, the star 1A, they have a normal hepatic influx of statins. But the other two, we can see star 5, star 15, they found to have a low or decreased transport efficacy of statin due to this genetic variation. So they are potentially can develop a myopathy. And another important factor is, as you remember, if you remember the first piece of this video, there are multiple statins, atorvastatin, rosuvastatin, simvastatin. So all of them are not going to be uptaken by SLC1B1. So that's where you have the interesting point. Like, you can switch to different statins, and you may get a better treatment outcome, avoiding the side effects. And most of these variants influence the simvastatin, which has a better treatment outcome, but it also has a serious side effect of uh, patients. So now I gave you two examples. I explained about the genetics today in a brief, and I'm giving you two examples about the influenced by the cytochrome P450 family and the statin, which is an anti-lipid drug. The lipid load, the adverse reaction of this drug is influenced by the genetic factors involved in the drug transport protein SLC1B1. So this is just a two. If you, there are plenty of explanations, plenty of drugs, in also the depression or epilepsy. There other, even the proton pump inhibitor also have a various side effects as well as. So they all have underlying genetic. Final section with genetic data sets. We know what is the, know how it's going to impact your therapeutic outcome, and how if you see the final video of this presentation.
And may result in significant side effects. Pharmacogenomics is the study of how our genes affect the way our bodies respond to medication. Because of our genetic makeup,
Second. Okay.
can save a life. Might help you and result in significant side effects. Pharmacogenomics is the study of our effects. Because of our genetic makeup, too slowly. The medication builds up in the body, effects. Other people's bodies break down the same at effective levels. There may be side effects from a medication. Can identify variations in specific genes, medications from the body. Using this information to predict whether a medication is likely to help you or hurt you. Pharmacogenomics is to provide the right drug for the right patient at the right dose. For example, if you have high cholesterol, describe the risk of a heart attack. But these drugs can also have significant side effects. Finding that out can be expensive. Time treatment. Pharmacogenomic tests how your body is likely to respond to a certain statin. That may help you, or even a different medication, to get the best results for you. Pharmacogenomic testing is available, which can help predict how an individual hundreds of different medications, including those used to treat heart disease, high blood pressure, psychiatric pain. Researchers are continuing to identify the body's response to specific is transforming the practice of health care by incorporating this information into your routine. Healthcare provider to learn more about pharmacogenomic testing.
Now we understand and how to move forward with the knowledge database we have with the genetic variation. What is a personalized drug choice? So you can see in the left side, that's a routine traditional treatment. You do a biochemical test, you go go to a doctor to measure your cholesterol levels or any other sugar levels in your plasma. A trial and error method. They give you the same prescription and same standard dose of a drug, which is given to most of the patients. And if you don't respond to the treatment, they may change the dose, they may change the drug, but the trend is going to change during the personalized medicine. In the right-hand side, you can see the biochemical test combined with the pharmacogenetic testing. Directly, the doctor understand and classify you based on your genetic makeup and as drug choice based on your genetic makeup and your biochemical value dose of the drug, or you may need a lower dose of drug or higher dose of drug or complete. It's going to happen very soon for most of the drugs because it's ultimately a therapeutic outcome should be a therapy. A drug should not be a poison. For example, a patient, those who need a drug, that going to cause a lot of side effects. And patients, those who need a high dose, the therapeutic strategy will not be efficient in them and par as you expect. And more importantly, the patient, those who have serious side effects, they are going to be the most important concerned patients we need to rule out. And if you can able to find it out early, you can avoid serious consequences of drug associated mortality or serious adverse drug reactions. So this is what we are moving forward in all the aspects of medication, not only restricted to the cardiovascular, in cancer, in other, even in antibiotics. So in central nervous system therapy, all those mode of treatments. And so this is a funny cartoon I wanted to show you. This is no more going to be in practice. Personalized medicine, can't you just take one of mine? That's a common cold or antibiotic. Just take one from my pill box. That is not going to happen in the near future because it's unique to have a special dosage, and we need to find it out. And it's going to be implemented soon, and it's growing, and that's what our future. And herewith I'm showing you some useful resources where you can able to find out about genetics and the CPIC. If you go to the Clinical Pharmacogenetic Implementation Consortium website, they have plenty of data about the genetic variations associated with the drugs, and you can read more. So this is for your own interest. And also the pharmacogenetic variation database, they have more information about some of the selective drug. If any one of you working in, even some of your family members may be taking some medication, that you can go check it out, and maybe you can try to figure out if you can able to find out any genetic testing available near your place, and you can able to do it you can talk to your about this to do preliminary testing that will help you to get a better treatment outcome. And with that, I would like to thank one and all for your kind patience and attention, and thank you for the opportunity to present with you guys, and it's kind of a great immense pleasure for me, and thank you. Now I'd like to take any questions or anything.
Yes, now we have come to the end of the session. Learn from yesterday, live for today, hope for tomorrow. So the important thing is not to stop questioning. So here we have a few questions have been posted in the YouTube live streaming, and I'll ask you only the few questions, and the rest of the questions will be sent through their reply to their email ID. The first question is, how can we determine the dosage level of any drug for a particular person if there is going to be genetic variation?
Okay, that's a very interesting question. So that is what the pharmacogenetic implementation consortium doing. So what they do, we know like the genetic variations, as I mentioned, in the metabolic enzyme or the transporters. So what they do, they create that specific variant in a cellular model, in an in vitro model, so cell culture model, and they try to figure out how much concentration of drug they effectively pump inside and out. And also they do a trial method. How they categorize based on their genetic makeup. So they just do the genetic testing previously administering the drug, and then they give them a drug for a couple of weeks and measure the peak plasma function and able to figure out how much dose might be effective in these patients, and they can able to get a clear picture about how much concentration or dose required for the patient. And if you read the PubMed publications from CPIC, that Clinical Pharmacogenetic Implementation Consortium, they method how they come out with this therapeutic studies.
Thank you, sir.
Any more questions?
Next question. What is your opinion on personalized medicine?
Can you repeat the question, please?
Yeah. What's your opinion on personalized medicine in your future? Is it possible?
You mean
Am I audible, sir?
the personal? Yeah, it's some glitch. You cannot
Yeah,
hear
personalized
the
medicine.
voice
Yeah.
clearly. It is definitely
Okay. What's
possible
your
because
opinion on personalized medicine? Okay.
yeah, because that's what that's what I'm like. We know like genetic information. We just had a bits and pieces of genetic document. They have all the information, and you see all thing. You can just do a whole thing yourself, $1,000. That means you can carry your genetic information in a pen. In future, what I think, imagine in another 10 years, it should be a few hundred technologies that are making it more cheaper in order to get knowledge of the genetic important genetic variations, how they are going to influence your health. I strongly believe in a decade or so, you will be having evaluate or screen your genetic makeup, and they will go for a decision of
The next question is whether can we cure cancer cancer by using crab.
So I asked whether we can cure cancer. After that, it stopped. So I couldn't.
By using crab, yeah, can we cure cancer?
That box like because it's hard to hear the question and read it
Sir, am I now audible?
better. Yes, you are audible now.
Can I ask another question? Yeah. Throw some light on success rate of plasma therapy.
Okay, plasma therapy is entirely different concept because everyone knows like now due to the COVID-19 pandemic, this infection comes in your body responds to that, and you produce the in your blood after the 1 week or time, the virus going to be getting rid of your body. Produce the amount of antibody to counteract virus. They need the plasma therapy. So plasma therapy is a successful therapy, but the thing is, the amount of virus what you have in your body and the ratio of the antibody in plasma should match to counteract to the virus. So the dosage, everything we cannot decide in those kind of therapy. Everything is a matter of fact. It's if you're lucky enough to have enough amount of antibody in the plasma, then definitely it may have a better impact. But ultimately, like you need volunteers to provide this amount of plasma who recovered immediately after this infection. So that's why people like it's not a feasible method of treatment for everybody, but it has a potential light in therapeutic response.
Thank you, sir. Another question. Is there any pharmacogenetic testing taking place clinically anywhere around the hospitals before prescribing a drug to a patient?
If you ask the things happening in India, there are pharmacogenetic testing centers available, but it is not a mandatory to do the testing since like we don't follow the FDA regulations as we do it in USA. In USA, if the FDA mandates a genetic testing before going with the therapeutic, starting a drug therapy, the physician must do the genetic testing, and with the results, they will start the therapy. But in India, it is not a mandate to do it, but the patient should have the self-awareness of understanding the pharmacogenetics. As a scientist, we all should know the importance of it to deliver this message to our family and friends to make sure there is an option of making sure your drug is going to work or not. So there will be genetic testing centers available, but it's not mandatory by government and other government agencies to do it before starting the therapy. It's everything now in the line of just trial and error method. But in future, it's possible, highly possible, like it may be following the same rules what before in United States of America. Some of the drugs definitely they need a genetic testing before administering to the patient.
Thank you, sir. A final question. What about the current status in India and how this could meet the demands of common man with these personal health disorders?
Very question like as I said, genetics is a very expensive subject because you cannot do genetic testing for a common man. For example, as I said, if you have few $1,000s, you can do your whole genome sequencing done, and most people can afford genetic testing in India is even you know the COVID-19 testing by real-time PCR itself cost a few thousand rupees. So it's not a cheap one. It's not an easy task to do it for a common man. But ultimately, the medicine is not going to differentiate between a wealthy person or a kind of lower level. Medicine should treat all of them in the same way. So all our dream is at one point of time we may get a free medicine, and that may help in getting all the better benefits to all the common as well as every human being in the world to get the better personalized therapy. So is highly expensive at the current stage, but in future it may go even lower so everyone can afford.
Thank you, sir. Thank you so much. In the chat box will be replied to your feedback link will be provided only
Ma'am,
at the end of
one
the
question,
session. So
ma'am. 1
kindly
minute.
fill the feedback form.
Yeah.
One question. Hello. Can you audible, sir?
Sir?
Yes.
Yeah.
Okay, sir, one question. Sir, what about the hydroxychloroquine in USA? What about the fact of usage of this drug, genetic variation or any other issue? Please.
Okay. So now the pandemic situation, like we want everyone want a wonder drug to just COVID-19. So now it is like we are in a it's a kind of war emergency. So war emergency, people will not do any trial. They will try doing and getting out the benefit out of it. So hydroxychloroquine, there are drug trials showing it may not be benefit. Also, it may have a lot of side effects in patients. But now the most of the researchers are focusing to find out the vaccine, medicine. But right now we don't have any other grip to come over COVID-19. Whereas there is the frontline prescription who have, because it is an anti-inflammatory drug, and most of the patients they have developed the cytokine storm due to the C cells, B cells, and macrophages in your lung, and that become the only way how we they are to go to the severe level of the disease inflammation. But not effects. But now people are doing some analysis how this may happen. But primarily they are trying to make use of this drug to improve the therapeutic outcome of the patient. But the question what you asked still need to address the large evidence and research.
Thank you,
Sir,
sir.
any other questions, sir? Any other questions from the participant side? Okay. Since there is no more questions, we have come to the end of the session. May I now invite Dr. S.R. Sendil Kumar, Head, Department of Botany, Center of Tiruchirappalli, to deliver the vote of thanks.
I think
Sir, I think there is some issue, sir.
Yes, sir. Sir, good evening
Yeah,
to
sir.
all.
Yeah, you are audible.
Yes, it is indeed a matter of pride for us that Reverend Dr. S. Peter S.J., our esteemed secretary, who felicitated the webinar with his noble words. Because of his moral support, we are able to achieve academic exercise frequently in the department. Thank you for the recent words. I say to Reverend Father Exami Xavier S.J., our permit me to mention our appreciation to the resource person, Dr. Vishnu Prabhu Raj Pandian, Center for Vascular and Inflammatory Diseases, University of Maryland, in our talk through various modes on genetic factors. Botany Department and on own behalf, I extend my most sincere thanks to you, Dr. Vishnu. We wish him for all your research and Dr. Sagai Sadish, Dean, School of Biological Sciences, St. Joseph's College, for his constant academic support. I take this opportunity to extend my sincere thanks to Dr. Sanusi, Assistant Professor of Botany Department, who made all arrangements. I congratulate them wholeheartedly. A special note to scholars, students, and other participants from our country, other international registered participants from Iraq, Saudi Arabia, Yemen, Morocco, Hong Kong, Indonesia, and Czech Republic for their interest and a bottom of heart to Dr. Kanickai Raj, Professor of Physics Department, platform for this webinar. Thank you, Professor. I hope you all had a wonderful informative webinar session from the Temple of Learning St. Joseph's. I wish you all
Thank you, sir. Thank you so much. Please kindly note, feedback link will not be sent individually. So please, so feedback link will be remain in the chat box. For do use the link shared in the chat box. Thank you.
Gratia plena,
Sancta